This is Charles. He was born a little bit different. Because of that, he didn't really fit in with other pigs at his barn, and he would often play by himself. His farmer wanted to take him to the doctor because he wanted to figure out what was wrong, because he didn't know what was happening. When he got there, the doctor actually said that the pig has something called piebald. Piebald is a mutation. Mutation is a change in the nucleotide sequence of an animal or human. Pigs are usually a pinkish brown color, but with the mutation, the pig has black spots all around it, like this pig has. The gene being changed is the KIT gene, which makes melanocytes, which make the color in the skin. The farmer thought that sounded really hard to understand. The doctor said, nope, it's not that hard, and said he had really easy to understand diagrams that would help him through it. Transcription is how DNA is transcribed in RNA. First, the enzyme helicase unzips the double helix so the process can begin. As you can see, the DNA template is being connected to nitrogenous bases on the mRNA strand. Adenine is the red one being shown, cytosine is the green one, guanine is the blue, thymine is the purple one, and uracil is the yellow one. This process takes place in the nucleus. As you can also see, thymine is connected to adenine, cytosine is connected to guanine, but in RNA, instead of thymine, it has uracil. What the RNA polymerase does is it helps make the RNA using the DNA template. As you can see, the RNA strand comes in and connects the DNA template to transcribe it into RNA.
As you can see in the second diagram, translation is the process of decoding the genetic information contained in messenger RNA, mRNA, to produce a particular sequence of amino acids, the building blocks of proteins. It is referred to as translation. It takes place in the ribosome where the rRNA is located. rRNA reads over the amino acids and links them together. Then the mRNA reads these amino acids and transfers them to the tRNA, which brings the amino acid and nucleotide chain to the ribosome. Translation is the process in which the genetic information encoded in a messenger RNA, mRNA, is decoded to produce a specific protein. This process occurs in ribosomes, cellular structures where protein synthesis takes place, codons, anticodons, and amino acids all play crucial roles in this process. Codons are sequences of 3 nucleotides in mRNA that correspond to specific amino acids or serve as start or stop signals for protein synthesis. There are 64 possible codons, but only 61 encode amino acids, while the remaining 3 functions as stop codons, signaling the termination of protein synthesis. Each codon specifies a particular amino acid or a signal for initiation or termination of translation. Next are the anticodons. They are sequences of 3 nucleotides found in transfer RNA molecules. These molecules carry specific amino acids to the ribosome during translation and recognize complementary codons in the mRNA. The anticodon of a tRNA molecule pairs with the codon in the mRNA through complementary base pairing. For example, if the mRNA codon is AUG, the corresponding tRNA anticodon is UAC, carrying the amino acid methionine. This complementary base pairing ensures that the correct amino acid is added to the growing polypeptide chain during translation. Lastly, the amino acids are the building blocks of protein. Each amino acid is specified by 1 or more codons in a genetic code. During translation, the ribosome reads mRNA codons and matches them with the corresponding tRNA molecules carrying the appropriate amino acids as the ribosomes move along the mRNA. It catalyzes the formation of peptide bonds between adjacent amino acids, forming a polypeptide chain. The sequence of amino acids in the polypeptide chain is determined by the sequence of codons in the mRNA, which in turn reflects the sequence of nucleotides in the DNA. In translation, the process of protein synthesis occurs in the ribosome and involves the step-by-step assembly of amino acids in a polypeptide chain according to the sequence of codons in the mRNA. The ribosome binds to the mRNA at the start codon, and the initiator tRNA carries the amino acids to it. Then the ribosome moves along the mRNA, reading each codon. Incoming tRNA molecules carry specific amino acids, bind to complementary codons in the mRNA. Peptide bonds form between adjacent amino acids, extending the polypeptide chain. The ribosome shifts along the mRNA, moving from codon to codon, and when a stop codon is reached, release factors bind to the ribosome, causing the release of the completed polypeptide chain. The ribosome dissociates and the protein is released. This process repeats for each mRNA molecule, generating proteins according to the sequence of codons in the mRNA. You can think of translation as typing a document. In transcription, DNA serves as the original template, much like a printed document. RNA polymerase acts as a typist, transcribing information from the DNA template onto an mRNA molecule, similar to typing a document on the computer, just as the document reflects the information from original source. mRNA carries the genetic information encoded in the DNA.
After the farmer learned all about that, he asked, "But what actually is piebald?" And the doctor said that piebald can be caused by either errors in DNA replication or from the damaging effects of mutagens such as chemicals, tobacco, and radiation, which react with the DNA and change the structures of individual nucleotides. Piebaldism is a condition characterized by the absence of cells called melanocytes in certain areas of the skin and hair. Melanocytes produce the pigment melanin, which contributes to hair, eye, and skin color. The absence of these leads to the patches of skin and hair that are lighter than normal. The reason for these absences is caused by the absence of the snail tube protein, which plays a role in the formation and survival of melanocytes. And then when they got home, all the little piggies played together, and Charles lived happily ever after.